Moving towards the last speaker of this second technical session, that is Dr. Priti Amritkar. She is director at EnviroCare Labs and has done PhD in chemical technology from ICT. She has been working in analytical domain over two decades, residue monitoring, nutritional analysis, fortified foods, measurement of uncertainty are her areas of active interest. She works closely with regulatory bodies such as BIS for standardization of test methods in Chemical, Food and Agricultural Department. She has many publications in national and international journals and has presented scientific and business lectures at many events across the globe. She is also a peer reviewer of the journal for AOAC International. Dr. Priti is empaneled as a technical assessor with NABL and is also a master trainer with FSSAI for pesticide and mycotoxin analysis in food. She is a recipient of many awards, including Scotch Award of Merit Award 2015, CII Award for Outstanding Performance Food Testing Laboratory Award 2018, and Outstanding Entrepreneur Award 2019. With this, we welcome Dr. Priti, and she will take us through the analytical requirements for import and export of soya products.
Thank you, Yogita, for the introduction. And I have been given the toughest task. I'm the last speaker, and I can see that the crowd is, kind of diminishing. But nevertheless, I hope that since you all have waited this long, I'll do my best and share my insights about analytical testing requirements. And just wanted to know how many of you are from testing background? The students? Only a couple of them, okay. So I can, be just relaxed and talk about it. No questions probably from you all. Okay, so can I get a mic? Yeah, so just want to share this slide with you. Thank you, Gita. So this is the slide. The reason that I have shown it here, I'll tell you. This is about some consignments from India getting rejected at EU countries. And the reason was that there were some peanut traces in the soybean. They were actually lecithin consignments, and they were rejected. Now, when you see this news, the importance of this news is that testing is important. So if you do not test your consignment properly, you are going to face problems. So this is the reason, and just to set the context of this presentation, I have shared this slide with you. Yeah. So this is about, I just wanted to draw your attention to the fact that analytical requirements will depend on the intended use of the product. So if the intended use of the product is about food, which is to be consumed by humans, the requirements, analytical requirements will be different. And if it is, for example, like soybean meal is hugely into animal feed, production. So the testing requirements for animal feed will be different. I just want to tell here that animal feed is a sector which is not really regulated in India, which was not regulated in India. And only recently FSSAI has come up with a directive saying that BIS, Bureau of Indian Standards, will now be making these licenses mandatory for the animal feed manufacturers. This is yet to be, I would say, come into full-fledged execution. But I just hope that this really works out, because this is important. Finally, the fish, even the milk and the meat that we all have, it is all coming from the animal feed that is being manufactured and fed to these animals. So this is also important. So if you just consider these soya products and FSSAI, these are the four major categories wherein all the soya products can be classified. These are mainly dairy products and analogs, fats and oils, fruits and vegetables, and cereals and cereal products. Now, how do you actually decide the analytical regulatory requirements? So when it comes to domestic, it is just the FSSAI that we have to follow. As a lab, I'm saying, I represent a lab, by the way. I represent a lab, as she said, and we are into testing from 1979. And food is a matrix which is really complex. and therefore a lot of effort also has to be done by the lab itself to standardize the methods. So for domestic and import and export, in fact, the testing requirements will be more or less similar. There would be slight difference when it comes to export, that we will see afterwards, but essentially domestic and import testing requirements will be very, very similar. The vertical standards, that is the vertical product standard, meaning FSSAI has categories for each product, and each product has been given a particular set of parameters that it needs to comply to. These are known as vertical standards or product standards. Like soya sauce will have a particular standard. Soybean oil will have a particular requirement. That is the vertical standard. And the horizontal standard means the safety standards. That is all across the food products. So basically it will be about contaminants like pesticide residues, or if it's a meat product, it will be antibiotic residues, toxins, metals, and other contaminants. So this is the difference between vertical and horizontal standards. When it comes to import, major thrust is always on the safety. So if you want to import soya products into India, the major thrust is always on the safety products like contaminants and melamine, etc. And sometimes it is also the label declaration if the label says a particular assay that needs to be done. For export, there is a very small amount that is exported from India to other countries, and majorly it is soybean seed itself. So when the consignment is sent outside, the value of the consignment in terms of tonnage is very high, and therefore weighment of that shipment is also very important, and a very small difference in the weighment also can be, can have a huge thrust on the importer and the exporter both. And the importing country, every importing country will have a specific requirement. Like China will have a different requirement, Japan will have a different requirement. So depending on what country you are exporting your product into, the requirements will change. Yeah, just wanted to also draw your attention to the fact that the analytical testing is mandatory for import, export, and for domestic, and it has to be carried out by a lab which is accredited by NABL, that is ISO 17025 accredited lab. And if it is for domestic import, it has to be recognized by FSSAI, and if it is for export, it has to be recognized by APIDA. Now, if you see the analytical methods, overall analytical methods, they fall into 4 categories. The first category is the physical test, that is actually just the physical examination of your product. Like soybean seeds, you actually have to examine the product and find out whether there are any insects, whether there are any extraneous matter, whether there are cracked seeds, immature seeds. So that is what is meant by the physical test. Chemical, generally what we do in the chemistry lab, fat, protein, moisture, those tests. And there is one test that is urease index value. This is particularly for soybean products. I'll come to that later. The third one is the residue, that is the horizontal standards I mentioned, safety standards. These include pesticides, aflatoxins, metal contaminants, and melamine. And the last but not the least is the microbiology aspect. So soybean seeds may not have any microbiological requirements, but the processed products will have some microbiological parameters that they need to comply to. And in addition to that, lastly I have written GMO. This is genetically modified organisms. Now when I say organisms, it does not really mean microorganisms, okay? It means plants here. In this case, it means plants. So these are the 4 basic methods that, we follow. Now this is just one example of a soybean seed sample. You can see there are shriveled seeds, immature green seeds, because these are immature since they are green. So as per FSSAI, there is a limit that is assigned to each of these categories, and the product needs to comply to these. This is only applicable for soybean seeds. This is another example wherein the product standard specification is mentioned, like soybean oil, for example. So oils, generally, all of the oils will have these parameters, but the specifications will change, like iodine value, sap value, unsap value, peroxide. All these are generally done for all kinds of oil, but soybean oil has a specific requirement, which is this. Fatty acid profile also is an important parameter for the oil. A lot of time, adulteration of the oil, when it is done, could be checked using fatty acid composition. The next is the pesticide, contaminant, and GMO group. Now, when it comes to the domestic and import testing of food products related to soya, for FSSR, there are 20 to 60 molecules which need to be tested for various soya products. And when it comes to EU exports, the list is very, very long. It's around 507 compounds that need to be tested ideally. I'm not saying that is what really happens, but ideally, this is what needs to happen. And for US-Canada exports, they have this national organic program. As per that program, there is a list of 186 pesticides which needs to be tested for every consignment that goes to US or Canada. Metals, melamine, aflatoxin, these are very typical FSSR requirements. For exports, they are not needed except for aflatoxins. And GMO, aflatoxins, by the way, are fungal toxins. They are produced when the soya products, basically seeds, they are exposed to a lot of moisture. The fungus is, if it is infected with fungus, these fungus, aspergillus fungus, will cause aflatoxins production, which is, aflatoxins are known to be carcinogenic basically, and they are very potent also. Therefore, aflatoxins need to be tested. Melamine, again, I don't know how many of you are aware of melamine controversy. There was a case in 2007 wherein, the China, some manufacturing company, they put melamine in the milk. Why? Because melamine is all about increasing the nitrogen content of the sample. So if there is a lot of protein, generally protein is estimated in terms of nitrogen. So when you put melamine, which is unauthorized actually, your nitrogen content and therefore your protein value seems to have increased. So people feel that there is a lot of protein in the sample, but that's not allowed. So melamine also needs to be checked, especially since we are talking about soya, there is a lot of protein already therein. So the last is the GMO. So for all export consignments, GMO is a must. European countries do not allow GMO food in their country, so GMO needs to be tested. And India also, as of now, there is no concrete, acceptance of the GMO food. US anyway allows, but still the consignment needs to be tested. This is just one glimpse of the pesticide residue analysis, just to give you a feel of what exactly is pesticide residue analysis. I mentioned that there are almost 500 to 700, even 800 molecules that have to be tested, depending on the importing country. So this is just one example. I have shown a method. This is just one of the methods. Since the number is very high, obviously one single method cannot address all the pesticide residues. There are a couple of more methods. There are almost 4 to 5 methods which will be able to address all of these pesticides. And therefore, there are different methods. I have just shown one method here. So once these pesticides are extracted, they are run into gas chromatography coupled with a mass spectroscopy technique. So there is a GC MS technique and there is a LC MS technique. So it may seem very easy that 500 molecules. No, it's a lot of work and it's a lot of hard work, I would say, that goes into it. Developing the methods, validating them, getting them under the accreditation. So this is about pesticide residues. And when there is no limit, or there is no MRL mention, the default LOQ, that is limit of quantitation, meaning in a consignment, if I am checking pesticides, each pesticide, I should be able to check at a level of 0.01 milligram per kg. That's the default MRL. Okay. The next is the microbiological analysis. As I said, soybean seeds, they won't have any microbiological requirement, but as the product is processed, various parameters have to be tested. For example, soya sauce, because it is fermented and it is a little complex product, the microbiological parameters are more here. Okay, so these also need to be tested. Generally, these microbiological parameters are tested using conventional methods. When I say conventional methods, they are all about long processes wherein you enrich the sample, then you cultivate the bacteria, then you do their biochemical tests, and it's a long procedure and it takes generally 5 days, 2 to 5 days, depending on which microorganism you are seeing. But nowadays, there are rapid methods available because today everyone wants everything fast. So the results also, the customers wants the results very fast. So rapid methods have been given a preference. I've just mentioned two of them here. The first one is the VIDAS and the second one is the RT-PCR. So VIDAS, you can see that it can detect the viable cells, but it takes 48 hours. Whereas RT-PCR is more of a DNA-based technique and can give you results within 24 hours. So imagine you have a batch of soya products, waiting to be released into the market. So instead of waiting for 5 days, when you use a conventional method, if you use a RT-PCR method, you can release your product into market just, after 24 hours. So that is the advantage of rapid method. Now we come to GMO regulation. I know Ratanji will be interested here. I'm sure about it. A lot of people are curious about GMO. So I can say that it is a very controversial subject, and nothing solid has been decided. Yes, but there is a draft regulation, and this was just brought in 2021. And two things here. First is that when we talk about GMO, it is not just about us. It is also about the nature, environment. And that is the reason Ministry of Environment and Forest and Climate Change, that is MOEF and CC, also is involved along with FSSAI in designing this draft. Okay, so there is a committee called GEAC committee, along with which this draft has been probably finalized or drafted. The first is the direct organism. Like for example, there is a direct plant like soya, which is GMO. This is a direct product that we will be using. So the first part is about those products. And the second one is about food or processed food containing genetically modified ingredients, like you use the soya, which is GMO soya, as an ingredient into something. And that product is the second category. Okay, so two different things. The product as such direct, and it being used as an ingredient. So when we talk about this, one thing is very clear, that the draft regulation has not permitted these GMO products to be used in the infant foods, because we do not know how GMO is going to affect in long run. It's relatively new, and only the time will prove whether they are good or bad for our health. So therefore infant foods have been excluded. Now the draft regulation says that GM food labeling. It says that all food products having individual genetically engineered ingredient, that is, say for example soya, which is GMO, 1% or more shall be labeled, and the labeling shall be, so the label will have, it contains GMO or ingredients derived from GMO. Okay. Now this is a catch there, that when you say 1% and above, it does not mean that 1% say jo kam hai, that is allowed. You have to understand here that every analytical method has a limit of quantitation. Okay? Probably 1%, because not many labs are able to, in fact there are no methods also currently, which can detect GMO less than 1%. And that is the reason it is 1%. Tomorrow as the technology evolves, as we have more sophisticated and sensitive instruments, RT-PCR or even qPCR, we may have this limit going even below at 0.1 or 0.01%. There is a program that US NGO, there is a NGO in US which runs a non-GMO program. Their requirement is testing of GMO at 0.01% quantitative level. So people are interested in checking at that level. So 1% does not mean that less than 1% is allowed. It is kind of taken for granted here that below 1% we will probably not be able to see the GMO. That's why. Yeah, so this is the GMO. I've just given a glimpse of it. Like basically our lab also is into GMO testing. And basically when you say GMO, there are two events that we check. One event is called a promoter event, which is called a 35S, and the second one is the TNOS. You have to understand that why do we need basically GMO food? Because, to make the crop resistant towards various infections or weather changes. Now, when you do that, you actually insert a plasmid DNA. And when you insert a DNA into the plant genome, there is a starting point and there is an ending point. So there is a promoter and the terminator. So these are most commonly used. Everywhere you see, these two will be used. So whenever in the soy product, if you see these terminators and promoters, you can say that it is a modified, genetically modified plant organism. So that is the reason these are seen. And we are also into quantitative testing. So recently we have, last year in fact, we got a request from NABL, that is the body that grants ISO 17025, to participate in a global PT, that is a proficiency testing program for quantitative analysis of GMO. So we have developed methods. We are waiting for the result. I just hope that we'll pass it. This is, so how the DNA is isolated, and how basically what is done in here is that the DNA is isolated, it is amplified, and then it is seen on RT-PCR. Okay, so this is how the result is obtained. So this is a positive control that I have shown here. So when you see the peaks there, these are the peaks that we should see, and the melt curve also is seen, how good the DNA has melted. So that is the melt curve. The last one is the vegan notification. Soya is very, very largely used in various vegan products. So the vegan regulation also was brought in very, very recently. It's not a regulation, it's just a notification with FSSAI saying that you can use vegan logo provided you comply to these conditions. So all of you are aware that no animal is used in the making of vegan, and therefore animal ingredients should not be present in your product. So essentially what you need to test is the presence of these animal ingredients which originate from animal origin, like milk, all dairy I mean, then there is fish, poultry, pork, and so on and so forth. And apart from this vegan, I would say regulation, the other regulation that the manufacturers have to follow is the packaging regulation. So there is a packaging regulation for FSSAI products. That also is applicable for vegan products. So you can't say that my product is vegan and therefore I don't have to follow the FSSAI packaging regulation. No. If you want to sell it in India, you also have to comply to the packaging regulation. Now what is this packaging regulation? Talks about basically a lot of times the food is packed in plastic containers. So plastic containers also leach out a lot of elements, and that is the test that is mandatory. So if you are packing your vegan food and selling it in India, you need to comply to these tests as well. And as I mentioned earlier, the allergens, which is milk, and the meat speciation, that if there is a meat, assuming that there is a meat, you try to find out what is the type of the meat. And that's the vegan logo that you have to use if you want to sell it in India. This is something that I just wanted to bring your attention to, because there was some concern from the soy milk industry, which was brought to the notice of AFSTI, and which is the Food Scientist Association. And that is the reason I have raised it here. There were a lot of manufacturers here. I'm sure they would also face this problem. So this is about basically a test called urease index in soy food products. This test has originated from animal feed industry. I'll tell you how this test came into existence. Conventional animal feed lacks an important essential amino acid called lysine. Therefore, to increase the nutritional value of the animal feed, soybean meal is added to conventional feed. Now, although soybean meal is a great source of various, nutritional components like proteins or even vitamins or fibers, it also has some anti-nutritional factors like trypsin inhibitor or lectins or phytates. Now, what do these anti-nutritional factors do? They reduce the nutritional value of the feed. For example, trypsin inhibitor. It inhibits the action of enzyme called trypsin. Now, trypsin is a proteolytic enzyme and is responsible for conversion of proteins to amino acids. Therefore, factors like these, they need to be removed from the meal. Now, let's see how soybean meal is produced. What happens is that soybeans are cracked, they're heated, they're flaked, and oil is removed by solvent extraction. And after oil removal, flakes are toasted and they're ground into meal. And during the above process, a mild temperature of say 90 degrees is applied to the meal. Now, why this heating? Heating is critical. Why? Because heating actually kills the anti-nutritional factors like trypsin inhibitor, and it also denatures the proteins and exposes new sites for enzymatic hydrolysis. But you also have to remember that you cannot overheat the soybean meal. Why? Because overheating will reduce the availability of lysine, because lysine is a heat labile amino acid, and therefore the feed manufacturers, they have a tough task. They cannot overheat the meal or they cannot underheat the meal, and therefore the protein quality, okay, they have to quantify the protein quality of soybean meal. Now, when you talk about this, it is difficult to measure these parameters, that is protein quality of the soybean meal. Okay, it is difficult to find out using a simple test. So what they have done is the soybean meal manufacturers, they have come up with an indirect way of checking the quality. They are correlating that to a test called urease index. Now, what is urease index and what it has to do with this? So I'll tell you, basically they are trying to only, because urease index, I would say urease is an enzyme again. So urease, destruction of urease by heating is highly correlated with the destruction of trypsin inhibitors and other anti-nutritional factors. So they assume that when these urease is killed, the anti-nutritional factors also will be killed. That is the logic. It's a very indirect method, but that's how it has been, and all the industry, all the, all while they have been using this method. So that is the reason this index, when it is between 0.05 to 0.2, they say it is optimally heated. And that is the reason they put this in the animal feed, that is soybean meal. But now this is for soybean meal, which is to be fed to animals, not to humans. Now what happens with humans? Do we not need lysine? Because this is all done for lysine basically, because soybean meal is a very rich source of lysine. And what happens with animals is that the conventional feed that they get is devoid of lysine. And therefore, the other animal feed is mixed with generally soybean meal. For humans, lysine is an essential amino acid. However, there are other sources of essential amino acids for lysine that we can have. We are not only dependent on soya. Okay? So that is the reason we may not, for human consumption, this test may not be required. Okay? So when this particular case was brought to our notice, we did write to FSSAI saying that they have to relook at this particular specification. Let us hope that we get a positive response from them. So this is about the analytical requirements, and would like to thank my team, Soumya, Neha, Moksha, Rahul, Dinkar, and Dr. Nilesh for helping me with the technical and regulatory insights. Would also like to thank USSEC for giving me this opportunity and to share my insights about the testing requirements. I would like to just end with one note. This whole conference was a beautiful experience, and a lot has been said about soy. Now, Marathi is my mother tongue, and in Marathi, soy means convenience. So really this product is going to be a very, very convenient product for all of us. You know, it's really a king of beans. They call soybean is a king of beans because very good quality protein at a very, very affordable rate. So cheers to soybean. Thank you.
Thank you so much, Dr. Priti. The entire presentation was giving a glimpse of what are the analytical requirements and how the methodology is being used in the laboratories. That has given a brief insight to all of us. Thank you for sharing it. May I request Dr. Ratan to please come on the dais and felicitate Dr. Priti.